This is the third video in the unit on Mendelian inheritance. In this video, we will discuss some of the complications that occur when trying to determine phenotype based on genotype. These complications arise because many traits do not follow the standard Mendelian properties of dominant and recessive alleles. Some of the complications we will address are alleles that show incomplete or codominance, along with some phenotypes where a single trait is controlled by multiple genes in a process called epistasis. We will also examine genetic crosses that involve a gene with more than just two alleles in the population. Oftentimes, when an organism is heterozygous, the phenotype they show is not complete dominance based on only one allele, but instead is either incomplete or codominance based on the interaction of the two different alleles. In incomplete dominance, the phenotype of the heterozygous genotype is intermediate between those of the homozygous genotypes. For example, in snapdragons, there is one allele that produces red pigment and another that does not, making the white flower. However, in the heterozygote, one copy of the red pigment producing allele does not make enough pigment to turn the flowers completely red. Instead, we get an intermediate phenotype of pink, which is a blending of the two allele colors. Notice that the alleles are no longer marked with uppercase and lowercase letters, because neither is dominant to the other. Instead, we use superscripts to denote which allele is which. So the CR designation is the red allele, while the CW designation is the color gene with the white allele. The genetic crosses are completed in the same way, as seen here in the Punnett square. But instead of getting 3 red to 1 white, like we would have with complete dominance, we instead see 1 red to 2 pink to 1 white. The phenotype ratio now matches the genotype ratio. Codominance is similar to incomplete dominance in that the heterozygous individual has a different phenotype than either of the homozygous individuals. However, the interaction of the two alleles in the heterozygote is different. In incomplete dominance, neither allele was dominant, so you saw a blending of the two phenotypes. Whereas in codominance, both alleles are dominant, so both alleles are observed in the phenotype of the heterozygote. We see here that in this flower, which instead of blending to pink, as in incomplete dominance, instead has red patches and white patches, as both allele colors are observed in the phenotype. Similar to incomplete dominance, upper and lowercase letters are not used since both alleles are dominant. Instead, we would again rely on superscripts to designate the alleles of the flower. A good example of codominance in humans is blood type. This is also a good example of a gene with more than 2 alleles in the population. Remember that even if there are more than 2 possible alleles for a gene, any one individual can still only have 2 alleles in each cell. For ABO blood type in humans, the blood type is controlled by 3 different alleles and can produce 4 different phenotypes. Blood type is based on the type of carbohydrate molecules you produce on the outside of your red blood cells. People with type A blood produce type A carbohydrate. The allele to make type A carbohydrate is the I superscript A allele. People with type B blood produce type B carbohydrate, controlled by the I superscript B allele. The IA allele and the IB allele are both completely dominant to the third recessive allele, designated lowercase I. This recessive I allele does not make any carbohydrate. People who are homozygous recessive for the lowercase I allele have O blood type. The last blood type is AB, people who produce both carbohydrate A and carbohydrate B on their blood cells. They are able to produce both because they have one A allele and one B allele, which interact in a codominant manner. Take note of the genotypes for each blood type listed in the table. People with type A blood can be homozygous for the A allele or heterozygous with an A allele and a lowercase I allele. The same is true for people with B blood. However, AB individuals must be heterozygous for the A and B allele, and type O individuals must be homozygous recessive. We will now work through a codominance practice problem. Read through this problem and pause the video. Try to complete the problem on your own and then hit play to watch my solution. This is a basic monohybrid cross. However, you now have multiple alleles and need to remember that the A and B alleles of blood type are codominant. This will affect how you determine phenotype, but do not affect the Punnett square of the actual genetic cross. First, we will draw our Punnett square and set up our gametes. In this case, the female individual can give an A allele or a recessive I allele, and the male can give an A allele or a B allele. We then fill out the fertilization inside the Punnett square. Looking at the Punnett square, we have 2 out of 4 possibilities to have a type A blood type. This is the same as 1/2. Out of the 4, we have 1 out of 4 that will have a type AB blood type, and 1 out of 4 that will have a type B blood type. The last complication we will look at is epistasis. Epistasis is when you have 2 genes, but they are interacting together to control the phenotype of only one trait. Because epistasis involves 2 genes, these are dihybrid genetic crosses. However, when you go to determine phenotype from the genotype, you will only have one trait you observing. The most common epistatic interaction is when one of the alleles of one gene masks the effect of a second gene. The gene that you see in the phenotype is the epistatic gene. Dominant epistasis is when the dominant allele of one gene hides the effect of a second gene, while recessive epistasis is when the recessive alleles of one gene hide the effect of a second gene. We will go over 2 examples of epistasis, blood type and eye color. Looking back at the blood types we just discussed, you will remember that there is an A carbohydrate, a B carbohydrate, and an O blood type that has no added carbohydrate. The key to epistatic nature of this trait is that there is a second gene, the H gene, which is necessary to make the H structure. The H structure is important because this is the structure that the carbohydrate is added to. The H structure is seen here. If you have an A allele, you make this type of A modification by adding this carbohydrate to the H structure. If you have a B allele, you make this modification by adding carbohydrate to the H structure. And again, the O blood type has no added carbohydrate to this structure. In order to add these carbohydrates, however, there must be an H structure to add the carbohydrate to. The dominant allele for the H structure gene allows the formation of the structure, while the recessive allele does not. The H structure gene is an example of recessive epistasis. If someone is homozygous recessive for the little h allele, they will not make the H structure. Without an H structure, this will mask whatever alleles they have at the ABO locus, because if there's no H structure, it does not matter if the cell makes A carbohydrates, there's nothing to attach them to. Without having an H structure, there is no carbohydrate attached, which would be the same as an O blood type. This was first documented in Bombay, India, which is why it is called the Bombay phenotype. Looking at this pedigree, we see that a male with type AB blood had a child with a female with type A blood. Their daughter tested as O blood type. Take a minute and think about why the dad was very angry when he found this out. Remember that to have an O phenotype, typically you must be homozygous recessive for the I allele. But that could not have been the case if the male was the father, because he only had an A or a B allele to give. This child was his, however. Instead, her genotype was A allele, little h, little h, which gave her an O phenotype even though she was homozygous for the A alleles. We will now do a practice problem on epistasis of blood type. Take a minute to read the problem and solve it on your own. Then watch my solution to check your answer. This problem can be done similarly to the other dihybrid crosses you learned using probability. The key will be to figure out how to determine phenotype from the genotype of the dihybrid. First, we will look at the ABO gene separately from the H gene. The woman can give an A allele or an I allele in her gametes, while the male only has B alleles to give. Filling out this Punnett square, we will find the probabilities for the ABO blood type. We then look at the H gene. Each individual is heterozygous, so they can each give a dominant H or a recessive H. Filling out the Punnett square will show us the possibilities for the genotypes that come with the H gene. After we have completed the Punnett squares, we need to go back to the problem to see what offspring phenotype we are calculating. The question asks for a child with AB blood type. To have an AB phenotype, the child will have to have an A allele and a B allele for the ABO gene because those alleles are codominant to each other. Child will also have to have a dominant H allele in order to see the ABO gene phenotype. Remember, if it was homozygous recessive for the H, then it would be an O phenotype no matter what the ABO gene said. Looking back at the Punnett square, we can see that there is a 2/4 chance of having the AB genotype and a 3/4 chance of having a genotype with at least one dominant H. The organism would have to have the AB genotype and the big H genotype. Since we say and, we multiply those numbers together, which gives us 6/16 or a 3/8 chance of having a child with an ABO blood type. Another good example of epistasis in humans is eye color. There are two genes that produce pigment for eye color. The dominant allele of the first gene produces brown eyes, and the recessive allele makes no pigment, leading to blue eyes. There is a second gene that has a dominant allele that produces green pigment, and a recessive allele that produces none. The brown-eyed gene is dominant epistatic to the green-eyed gene. If you have the dominant brown allele, it does not matter what green alleles you have in the green gene. Because the brown pigment is darker than green, so phenotypically the eyes appear brown whether you produce green pigment or not. However, if you are homozygous recessive for the brown-eyed gene, so you are little b, then it does matter what the second gene says. If the genotype of the second gene contains a dominant allele, then you will produce green color leading to green eyes. But if the second gene is also homozygous recessive, then you will still produce no pigment leading to blue eyes. Along with various interactions between genes and alleles of the genome, phenotype is also affected by the environment. One example is coat color in Siamese cats. Siamese cats have a gene that encodes an enzyme to make a dark pigment. However, the enzyme is temperature sensitive. It only works in colder environments. This is what gives Siamese cats their standard patterning of dark ears, feet, and tail. The extremities are colder than the core of the body. Because the enzyme is temperature sensitive, cats who live in an overall warmer climate will have less coloring, and those who live in colder climates will have darker coloring. Additionally, you can see this environmental effect in action. If a Siamese cat needs to be shaved for surgery, and then you need to have a cold compress on that area to keep the swelling down, the cold compress will lower the temperature of the hair follicles in that area. This will allow the enzyme to be active, and the portion of hair that grows back will be darker in color than the rest of the cat.